Hello learners, welcome to the session of the Introduction to Optical Microscopy Theory and Practices. So in this session I will teach you about the basic principle and theory of the optical microscope. So this is the topic of the B.Sc Honors Biochemistry program, and the course is cell biology. The course code of this course is the BBCCT 103 block 1. So today's session I will focus on the different kind of the optical microscope and what is the principle and application of the optical microscope. Have you seen this slide like microscopic images? Of course you might have seen this kind of the different kind of microscopic images under microscope in your biology courses at undergraduate level and the 10+2 biology laboratory. So these are the microscopic images which is captured by the microscope. So microscope is enabled to us to view of the smaller things that cannot be viewed by the human eye. So because these kind of the microorganism, cells, subcellular organelles are microscopic in size. So we need a microscope to visualize the smaller things. In surrounding us there is an invisible world, and this invisible world contains smaller things, a different kind of the microorganism, bacteria, viruses, and even the cells. And our body is made up of the different kind of the cells, but we cannot see the cells as we can see our body, and we can see animal and plants around us, but we cannot see the basic structure of the cells through our eye. So we need a microscope. So in this session I will go to discuss about the basic principle and application, how microscope is used and how can we utilize the microscope to view of the smaller things. So in this picture you can see different kind of the plant cells, human cells, red blood cells, bacteria, and even viruses. So this kind of the micro images captured by the microscope. So microscope is very important things. So in this session I will cover the different kind of the topics which is related to the microscopes, like origin of microscope and its uses, basic parts of the microscope, how microscope is working, basically the working principle of a microscope, how image is formed in the microscope, and what are the important parameters of the microscope like magnification, resolution, and resolving power, and numerical aperture, and the types of microscope. And in the end of this session I will talk about what are the safety precautions during the microscopic observation. So the first question is arise in your mind that what is microscope? Micro means the small things and scope means to look at. So the microscope is the laboratory device that can magnify and resolve of the smaller things. So this is kind of the microscope. Microscope is a laboratory instrument that visualize the smaller things that cannot be viewed by the human eye. So it is become of the instrumental instrument in the science. So it is based on the interaction of the light, biological specimen, optical lenses. And there are three important parameters here we want to discuss here. Basically the microscope is based on the magnification and resolution and the contrast. So why microscope is important? Basically the teaching and research, and at the level of the microscopic level of the biological specimen we need a microscope. So microscope is an instrumental instrument that can help to study of the biological things that cannot seen. So the cells, living organism like the bacteria, subcellular organelles, DNA, proteins, and at the subatomic level the microscope enable us to see and examine the various kind of the cellular structures. So in a scientific process microscope has been developing a wide range of the microscopic techniques like bright field microscope, fluorescence microscope, and there are different kind of the microscope you can see in this slide: light microscope, fluorescence microscope, electron microscope, stereo microscope, confocal microscope, atomic force microscope, inverted microscope, and retinal imaging microscope. So but basic principle is the same. So in the microscope is also enable us to diagnose the diseases. So it is a current technique which is utilized in the advanced medical imaging system. So first we start what is the history of the microscope. So basically Jacques Janssen in 1590 he utilized the two lenses and fitted into the a tube, and he developed the first simple microscope. And further the Robert Hooke, which is the well-known scientist as a cell biologist, he used the first combined microscope to visualize of the dead cork cell wall, and he coined the dead cork cell wall like the cells and the honeycomb. So, and further Antoine Leeuwenhoek, he was not a scientist by profession, but he is the cloth making person. So he first utilized the simple microscope by improving the quality of the lens, and he first time visualized the living organism like red blood cells and the bacteria like that. So in this process of the journey of microscopic development, these three kind of the scientists who led the microscopic field. So in microscopy we used to study the living organism at biochemical soil level using the microscope. So in microscope there are two kind of the microscope we generally used. You know, the microscope is based on the two types of the beam of the matter, beam of the electrons or the light. So in microscope is of two types, like light microscope and electron microscope. In light microscopy we use the light to visualize of the biological samples, and in electron microscope we utilize the electron beam to visualize of the subatomic particles or the smaller things. So these two kind of the microscope is different based on the utilization of the source of the light. So in this session I will talk about the light microscopy. So in light microscopy generally three kind of the things are necessary. First is the visible light is used, optical lenses and the specimen slides. So first I will talk about what are the basic parts of the compound microscope. So first is the eyepiece. So eyepiece is fitted to the top of the microscope, and then the revolving nose, which is called the nose piece, which utilized for the movement of the optical lens. So under the revolving piece there is a certain kind of objective lens. 3 to 4 object lens is there, and then condenser lens which condense the light, and diaphragm which increase or decrease the intensity of the light. How much light is entered into the object lens is decided by the condenser lens, and the diaphragm and the light source. So light source basically the halogen lamp or tungsten lamp. It is an artificial source of light which illuminate the light up to the diaphragm, condenser, object lens, revolving doors. So in this process of the basic component of the microscope, the other kind of the parts is here, like you can see the arms, and the arm is the hold the microscope, helps in the hold in the microscope. It the coarse focus knob. So coarse focus knob is used to help to the up and down of the objective lens near to the specimen stage, and fine focus knob is help to the sharp of the object of the specimen, and stage adjustment knob is basically movement of the slide near to the objective lens and the specimen slide, and the 11th part is the light control knob, is help to increase or decrease the light intensity which is at the level of the light source. So these are the 11 kind of the basic parts of the microscope. You can see here the first the eyepiece. Eyepiece helps to visualize of the biological specimen which look into the at the level of the objective lens. So basically three kind of the microscope is three kind of the parts is very important in optical microscopy. First the objective lens. So objective lens is the most important part of the optical microscope, because the optical lens is first form the image of the specimen. So here there are three kind of the objective lens are used, like 4x, 10x, 40x and 100x. So basically what does the objective lens here? It collects light coming from the specimen, the various kind of the specimen, and produce a real and inverted magnified image. So you can see in this figure there are objective lens. So objective lens is not a single lens, it is multiple lens fitted in the objective lens. And second is the eyepiece. So eyepiece is very important. It further magnified the image of the formed by the optical lens. So the eyepiece is the most important lens through which we look a specimen under the microscope. What we see, the real image of the specimen, is basically through which the eyepiece. So it is generally 10x magnification. This is important to remember when calculating the total magnification of the image. So earlier we have talked about that there are two kind of the things here, resolution and the magnification, which is the important parameter of the microscope here. Another, the specimen slides. Basically what we observe, which is the specimen slides. Basically when we put the specimen on the slide, we have to prepare the specimen slide. So there is another method how to prepare the slide. So you can see here, this is the specimen slide which is placed in the specimen stage on the microscopic instrument. So now I talked about the pathway of light. What is the pathway of light in optical microscope? You know, in light microscope, light is used. Basically sunlight can be used here, and or the artificial light like halogen lamp can be used to visualization of the biological sample. So here you can see the light source coming from the light source, and it falls on the mirror side, and the mirror again reflect this light into the condenser lens. So and condenser lens focus the light up to the specimen, and then it interacts with the specimen. Various parts of the specimen interact with the light which comes from the condenser lens, and after interaction of this light with the specimen matter, then it goes to the objective lens, and then objective lens visualize and magnify the image of the specimen which is inverted, and then it further goes into the eyepiece, and through eyepiece we can see the biological specimen magnified image through our eye. So this is the pathway of light. So in the process of the image formation, the light alignment should be matched with the optical lenses. Here you can see the condenser lens, the objective lens, and the eyepiece. So there are three kind of the lenses are necessary in proper alignment through which the light passes and the visualize of the biological image of the specimen. So here we have to remember that the biological specimen is very smaller in things when we prepare the biological slide. Next question is can arise in our mind: how is image formed in light microscope? So you can see here the light source and the objective lens and eyepiece. So the magnified image is first formed by the objective lens, and what we see, the image of the biological specimen is real and inverted, and it enlarged. So basically the properties of the light is more responsible with the interaction of the specific matter and the property of the optical lens. So this image is formed at the intermediate image plane inside the microscopic tube. So you can see this is the object here. This is the objective here. Object means the biological specimen that to be observed under the microscope. So this is the objective object and objective lens, and when light interacts with the object, it further interact with the object of the matter, and further it enter into the objective lens and the eyepiece. So the interaction between the objective lens and the eyepiece, and the image is formed in the magnified image in larger view. So the final image is inverted, means it is inverted means the light is interact with the matter, and it create the not a right view of the specimen, but it inverted in with the magnified view of the biological specimen. So how light interact with the matter? It means basically when the light interact with the specimen matter, it can be absorbed, it can be reflected, it can be refracted. So the basic properties of light is very amazing. So what we see in the microscope, or what we see in the surrounding it. So when we take an example, when we suppose this is the glass and it interacts with the matter. So when light interacts with matter, some light passes through this glass lens. It can transmit without reflection, and some light can be reflected from this side, the glass lenses here. So basically our eyeglasses helps into magnify of the images when our eyesight is big. So this is the kind of the glass properties here. So we can see the magnified image when our eye is big. So this kind of the property is utilized in the microscope. So the microscope has two kind of the lens, the optical lens and the eyepiece. So first image is formed by the optical lens. So when light interact with the specimen, it can be absorbed by the specimen matter. It can be back again scattered with the interaction of the specimen, and it can be 100% transmitted through the specimen matter. So this kind of the properties are utilized for visualization of the biological sample in the light microscopy. So here you can see that this is the sunlight. Light interact with the matter. It can be transmitted, it can be reflected, and it can be refraction. What we see here, that it leads to the property of light with the interaction of the matter. It leads to the working principle of the microscope. So when a ray of light passes from one medium to another medium, it is called the refraction. So in light microscopy, refraction is more important than the transmission and the absorption of the light. So here, what is refraction property of the light? So when light enter into the one medium to another medium, the direction of light is totally different. The speed of light can be same, but direction, the pathway of light is slightly deviated from it goes to the one medium to another medium. So suppose we take one example here, that this is the slide. This is the slide here. You can see I take this slide here. This is the microscopic slide, and when we put the small dots here, so this is kind of our, just for I take example of this dot of this ink here. So what will happen when I utilize this one of the light source of this, when we interact? So I'm giving the example of this. When this light passes through this, some light, it ultimately crosses this light, and some of the light rays interact with the matter. So this blue spot, ink spot, can be absorbed some of the light, can be refracted of the light. So when light interact with the glass, after passing the glass, light is deflected. The path of the light is different. So this is the kind of the properties which happen under the light microscopes, and this is the process. Then image is formed in the light microscope. So this bending properties of light, when light goes from the air to glass medium and from the glass medium to the water, so the direction of light is different. So in this process, the refraction, the degree of the refraction is called the refractive index. So this is one of the example here. Then we take the refraction properties of light, which is more important in light microscopy. So light goes to the glass and can enter the water and comes back into the air. So the light angle is different. So this kind of bending property is very useful for visualization of the smaller things. So light bending of light determines the refractiveness of the medium, because it enter into the different kind of the density of the biological specimen. If material is more dense, the light will slowly move. If the lighter density of the medium, it speed up the light process enter into the different medium. So what are the microscopic characteristics? So what are the different kind of the parameters we take the consideration while formation of the light microscope. So in order to generate good quality of image in the light microscopy, we generally consider the three kind of the parameters here. First is the magnification, second is the resolution, and third is the contrast. So now I discuss in one by one. So in the magnification is the process of the enlarging an object or specimen under the light microscope. So generally magnification is a combination of the optical lens and the eyepiece. So you know that in light microscopy the three kind of the lenses is used. Optical lenses are used, three kind: 4x, 10x and 40x, and eyepiece generally is of 10x. So when we calculating the magnification of the light microscopy or magnification of the biological specimen that look into the under the light microscope, suppose if we use the 40x objective lens or the 10x ocular lens, the total magnification of the microscope is the 400x. So I am giving just example. So in this table you can see the magnification objective lens 4x, 10x, 40x, and eyepiece is similar to the 10x. So when total magnification, when we use the object lens 4x and eyepiece 10x, the 40x magnification of the microscopic image, 10x multiplied 10x is the 100x magnification of microscopic image, and when we use high power of optical lens 40x and multiply 10x, so it is the 400 magnification. So in light microscopy generally it can produce the 1000x to 2000x magnification. And another point is the resolution resolving power. So it is very important. The resolution of microscope of lens is typically described the smallest distinct between 2 points that can still be distinguished as a separate entities. So in a small biological specimen, there are different kind of the structure in biological specimen, or we can say the object that can be visualized under the microscope. So the microscope has an ability to distinguish two close points as a separate entity, which is called the resolution. So resolution is very important. So a smaller value of the resolution, it increase the resolving power and image clarity and detail. So you can see in this figure, then B figure is the clearly resolved, but A and C figure is not clearly resolved by the microscopes. It also indicate that the quality of the microscope that can be resolved the two things as a separate entity. So in light microscope it has the limit of the resolution. So resolving power of the human eye is the 0.20 mm. So human eye cannot see less than 0.22 mm biological specimen or the cell structures. So resolving power compound microscope is the 0.20 micrometer. So the light microscope has an ability to see the microscopic object that can be smaller at the 0.20 micro in meter, and electron microscope has a better resolving power, which is the 0.001 nanometer, because the electrons are very smaller as compared to the light microscope. So I come to the resolving power. So resolving power is the ability of the optical component of microscope that visualize the two closely pointed as separate entities. So the resolving power microscope depends on the wavelength of light and sin alpha. So this is the formula you can see here. Resolving power D is equal to 0.612 lambda upon n sin alpha. So in this formula, lambda is the wavelength of light and 0.612 lambda is a constant. D is the minimum distance to distinguish between two close points within a specimen, and sin alpha is the numerical aperture. So the shorter wavelength gives the higher resolving power. And now come to the numerical aperture. So numerical aperture is represented by the NA. NA is a measure of the lens ability to gathering light, and the higher numerical aperture, the better the resolution. So n is equal to n sin alpha, that we have talked in last slide. N is the refractive index of the medium. Sin alpha is the half angle of the light rays entering between the object and the objective lens. So sin alpha is defined as the when we the light angle, the half angle of the light between the specimen and the objective lens. So this is kind of the numerical aperture that is formed when we objective and the specimen is near about. So the angle of the numerical aperture between will be higher. If the distance between the specimen and object lens is higher, increases, then numerical aperture angle will be the shorter. So in that case, we can increase the numerical aperture by using the different kind of medium. One of the medium is the oil immersion medium, which has the higher refractive index 1.5. Then air has the 1 refractive index, and water has the 1.33 refractive index. So we can increase the numerical aperture by using the external medium put into the mix with the biological specimen. So higher N value increase the resolution. So in this method we can increase the resolution by increasing the numerical aperture, and we can also utilize the short wavelength of light, which can be also helpful to increase the resolution of the microscopic images. So refractive index, I talked about already that refractive index is the fundamental property of the lens that define the light travel. So when light enter into one medium to another medium, it refracted. Its pathway is slightly different when it goes to the air to the glass slide and glass slide to the air. So here you can see the white light is diffracted and transparent medium and another reflected. So this kind of the deflection produce a inverted image under the microscope. So this is the reason, that's why we get a inverted image under the light microscope that can be viewed by the eyepiece. So now come to the types of compound microscope. So we have learned about the fundamental properties of light, working principle of light microscope, and what are the important parameters of the light microscope, which are the magnification, resolution, resolving power, and the numerical aperture. And most important property is the refraction or refractive index of the light. So there are many types of the light microscope we are using nowadays in laboratory, but some of the common microscope are bright field microscope, dark field microscope, phase contrast microscope, fluorescence microscope, and confocal microscope. So this kind of the microscope are available in the science labs. So I talk about only two microscope here, bright field and dark field microscope. So the bright field microscope, basically bright field, at its name bright field. So its image is dark against the bright background. So what will happen when the light passes through the objective lens? Basically light interact with the specimen and it transmitted. So transmitted light is captured by the objective lens and it further magnified by the eyepiece. So here you can see the different kind of the plant cells and bacteria are visualized here. So the dark images appear against the bright background. So you can see the bacteria and the plant cells here we are looking. And basically some of the specimen are stained, some of them are not. So a bright field microscope require the staining procedure to visualize of the biological specimen. So before preparation of the biological specimen, we generally stain of the biological objects, then can be visualized under the bright field microscope. It is a common type of bright field microscope, is generally used in the microbiology to visualize all the different kind of bacteria, gram positive bacteria and gram negative bacteria. And it also is very useful for the cell biology to visualize the different kind of the cells and the nucleus. Another microscope is the dark field microscope, as its name, dark field. So it produces the bright image of bright image of the specimen against the dark background. So what happens? In this microscopy, we generally stop some kind of the light by inserting the condenser page stop in between the source and the in the condenser and the specimen in between. So light is scattered from the specimen into the objective and producing the bright image of the biological specimen against dark background. So you can see here different kind of the bacteria is brightly illuminated against the dark background. So then these are the precaution while using the microscope: properly handling, cleaning. We generally use a lens paper for cleaning the microscope, always stored and covered. Stage and specimen, basically use the carefully. We put the biological specimen at the stage of microscope. Focus, adjustment, and proper power supply. These are the common precaution for using of the microscope. So this kind of the summary. Now you understood the different kind of microscope, what the principle and application, magnification, resolution, refractive index, like that. And this session might be helpful to understand the basic principle and theory of the microscope. So this is some kind of practice question. You can solve the labeling the microscope parts, and you can calculate the different kind of the magnification if you are using different kind of objective lens and the eyepieces. So you can download this course from our EGNOU course portal at the BSCBCH section. These are the suggested readings, and thank you.